Hello, welcome to you all. I'm very happy to welcome you to this webinar about the role of iodine in plants. I'm also very happy to welcome Dr. Katja Hora. Dr. Hora has been involved both in the scientific world, discovering more knowledge about the role of iodine in plants, as well in the research to provide a solution and a product that is able to apply iodine for plants in an easy and safe and adequate way. But before I give the word to Katja, I would like just to mention a practical thing, and that is that you are able to point out any question that you might have during this session in the chat room. And then we will take them into account and probably come back to them at the end of the session or eventually later. Also in this chat room, you will be able to download some documentation, and the presentation, of course, will be available for you after the session. But now, Katja, the floor is yours to help us understand more about the role of iodine in plant nutrition.
Thank you, Koen, for the introduction, and indeed, I'm glad to be able to explain to you why iodine is needed by plants. I'm going to start with an introduction on iodine, and then I'm moving on to the fundamental science behind iodine as a plant micronutrient based on phenotypic, genomic, and proteomic studies. And Koen will then continue to explain how to apply the right dose of iodine in fertigation. So, introducing iodine. Iodine is an essential nutrient for humans and animals. It regulates the production of hormones in our thyroid glands and is part of thyroid hormones. Iodine binds covalently to tyrosine, and that makes our hormones. You can have a mild iodine deficiency, which originates from not having enough iodine in your diet. And this slows down your thyroid function. And in pregnant women, it's especially dangerous because it can cause irreversible brain damage to children. Iodine deficiency in humans is widespread, and this is why there are a number of solutions to prevent iodine deficiencies in humans and animals, such as fortification of salt, where table salt is fortified by adding iodine to it, mineral supplements, and iodine is also given to farm animals in salt lake blocks. So, but what about plants? We ask ourselves three questions. Do plants also need iodine? Can plants also suffer from iodine deficiency? And if yes, how can we prevent iodine deficiency in plants? So this brings me to the fundamental science. The main evidence for a nutritional role of iodine in plants was published in 2021 in this article in the Frontiers of Plant Science, which you can download for free. The work was performed by scientists at the Scuola Superiore Sant'Anna in Pisa, Italy, and of the National Research Council in Naples. And also myself and my colleague Harmen Challing-Holverda were involved in this research. The main conclusions are that plants have a nutritional role in plants. And the research was carried out on this little plant, which is called Arabidopsis thaliana. It's a very small plant. It grows very fast, and this is why it's a very well-known model organism to study plant physiology on. All the genes are known, and also it's understood how the proteins in the plants work together to make it grow. The paper brings three key messages. First, there is a study on phenotype. Can you see that plants suffer from iodine deficiency? The second is on the level of gene expression, where we show that plants react specifically after perception of iodine in a nutrient solution, and that this perception is indeed unique for iodine. And the third one, the most exciting one, is iodination of proteins. It was discovered that plants bind iodine and protein similar to how humans do, with important and conserved function in the biological processes of plants. So I will summarize these 3 points for you. First, the phenotype. What did we see in plants? How can a plant be iodine deficient? Well, this trial was done with Arabidopsis thaliana grown in a nutrient solution, so grown in water with nutrients in it. On the left, you see a picture where the plants were iodine deficient, and this was done by using ultra pure water and ultra pure salts. So the iodine content in that water was below the limit of detection of the equipment. And only 4 weeks after planting, you see that these plants have hardly yet started to develop flowers. The flowers are on the stems of these plants. On the right, you see two pictures where the same plants grown at the same time in the same climate room were provided with either 0.2 micromole per liter of iodine in the water or 10 micromole. And you see these plants flower much sooner, also have more flowers, more seeds, and also reached higher biomasses. So indeed, this shows that plants can suffer from iodine deficiency, and it shows as smaller plants retarded flowering and a lower seed production. Not only is this seen in the above ground part, but you can also see this in root growth. Well, this part was not published, but what you see here is Arabidopsis thaliana seedlings grown in an in vitro medium. And this is a similar medium what is used for the propagation of plants in vitro. Now, normally iodine is added to those media, so a normal medium contains already 5 micromole of iodine per liter. And you see on the left, if you take it out, what remains in the medium is 0.6 micromole of iodine per liter, and the roots are much shorter without iodine. In the optimal range, between 5 and 10 micromole per liter, the roots grow longer, but also it was visible that they develop more lateral roots. Not so much in this picture, but it was well observed. It's also possible to apply an excess of iodine, as you see on the far right, where the leaves of plants are becoming a bit chlorotic and the roots develop less well. But remember, this was 10 times more than the upper dose, which we would consider the optimal range for iodine. So the second part, modulation of gene expression. This was investigated by growing these small Arabidopsis seedlings in these wells with nutrient solution, and they were exposed for only 48 hours to either sodium iodide, potassium iodide, or potassium bromide. The control was iodine deficient. And the resulting gene expression was analyzed using a gene chip technology and data processing and analysis. We included bromide in this comparison because bromide is a sister species to iodine. They have very similar chemical properties. So we raised the question if the plant would recognize bromide as it recognizes iodine. Well, the answer is no. We found a clear evidence of gene expression. This picture shows gene expression in the roots. And it was unique for the presence of iodine. If you look at these circles, the number of genes in bold in these circles are the genes that respond only to iodine. So not to sodium, not to potassium, and also not to bromide. As you can see in the bar below the picture, which is very vaguely red and blue. And this means that the genes, and every line there is one gene, these genes did not respond to bromide. Well, if you compare it in contrast to the expression which was found by exposing these plants for only 48 hours to potassium iodide and sodium iodide, this is immense. In the shoots, similar things happened, only here there were less genes that expressed, but this is correlated to the only short time of exposure of these plants. So also, these plants were supplied this iodine in a micromolar dose, so a dose similar to a micronutrient. So what does this tell us? It tells us that a plant recognizes iodine and responds with a unique activation of genes in the micromolar concentration. Now, because it's Arabidopsis, the identity of these genes is well investigated, so it could be analyzed how iodine is involved in the plant's metabolism. Now, the outcome of that analysis was that iodine is involved in communication between cells and plant organs and in the response of the plant to changes in the environment. And this can be a response to stress signals such as high temperature, drought, salinity, or low light. And an important aspect of this response of the plant is called calcium-dependent signaling, and that is comparable to the nervous system of people. So this is the way a plant recognizes its surroundings. And also iodine showed a clear expression in the genes involved in calcium-dependent signaling. So brings me to the third part of the evidence, the iodination of plant proteins. This was done using an artificial isotope of iodine, iodine 125. And normally iodine is not radioactive, but specifically for this experiment, it was made radioactive. So the scientists could follow what happens with this iodine when you put it into the nutrient solution and then analyze what happens to it in the plants. Now, to find back proteins which contain this radioactive iodine, a technology called autoradiography was used. And this is an example to explain to you how the results look. If you look at the black band where this red arrow points at, this band means that the radioisotope is bound by the plant to a protein between 75 and 100 kilodalton of weight. Important to notice that this black band can contain multiple proteins to which iodine is bound. Now, these are the results of the experiment with Arabidopsis. You see a number of black bands, and these are all the proteins which have iodine bound to them by the plant. Now, this was not only done with Arabidopsis, but also in 4 different species of crops: maize, tomato, wheat, and lettuce. And you see that these black bands also appear in these crops after they've been given the radioactive iodine, which means that the role of iodine in plants may be conserved in evolution, and it's a general way that plants use iodine. Now, also these 82 iodinated proteins could be identified with still another method. And the outcome of that analysis was that these proteins, and there are 40 of them that were identified in the roots and 42 in the shoots of the plants, and they're involved in photosynthesis, in growth regulation, in energy and protein production, in recycling of elements, assimilation, and the plant's response to stress. Now, so to answer with these 3 questions, do plants also need iodine? For that, we answer a yes. And iodine is now classified as a nutrient with proven benefits for plants, as you can see in this infographic of the International Fertilizer Association, which includes iodine in 19 plant nutrients needed by plants. The second question, can plants also suffer from iodine deficiency? Well, that's also a yes, as you can see clearly in this picture and also in the roots. So if yes, how can we prevent iodine deficiency in plants? And a related question, is it likely that crops are also iodine deficient? Well, we saw from these experiments that also commercial crops contain iodinated proteins. But is there a likelihood that they are deficient in practice? Well, for that, we look at the parallel to humans. We know that iodine deficiency in humans is endemic in Europe because they lack iodine in their diet. And only recently, a study, EU thyroid, was done, which estimated that 50% of Europe, of the infants in Europe may be at risk of being iodine deficient. And this is because of a lack of iodine in the diet. So what about crops? Can we also expect to find iodine deficiency in crops? Now, of course, we set out to find out. And this picture represents the average value of iodine in different types of water collected from more than 100 agricultural production farms worldwide. We collected well water, tap water, surface water, and the nutrient solution. And you see on average in these samples, there's less than 1 micromole of iodine per liter. And in many of these samples, the level was below 0.2 micromole per liter, as you saw was the lowest dose in the Arabidopsis experiment. So this is a problem, but is there also a solution? And for that, I give the floor to Koen.
Thank you, Katja. Well, in your introduction, you already mentioned that there exist programs for fortification of iodine in food and feed. So, in agricultural, this already indicates that in agricultural crops, we are expecting already a shortage in iodine. Otherwise, we wouldn't have not have all these European programs for fortification of iodine for humans. But if it comes specifically down to plants, of course, it makes sense to really study the levels that are available for plants. And the first indication was already done by a study from 2015 by Fuchs and Johnson, who describes the levels that we encounter of iodine in different water sources, in sources in nature in general. And this already indicates that usually we are far below the 1 to 10 micromole per liter that we would uh, consider as adequate levels. More precisely, the 100 sample evaluation of 2019 and 2020 you just referred to really shows, in comparison to what we would call the indicative range of adequate levels of iodine in a nutrient solution, shows really that we are still well below this 1 to 10 micromole per liter range. I give some more specific examples of European references um, that even show that often we are below 0.2 micromole per liter in many cases. A first example is an analysis of a representative drip solution in the Westland in the Netherlands. This is close to the coastal area, even where we would expect maybe some higher iodine levels. But look, when we really do the analysis, we see that we measure levels below 0.05 micromole per liter. Another study of 12 water samples in the Athens area in Greece, and this area is known to be an area where endemic iodine deficiency in people was rather low, but this showed averages of 0.04 micromole per liter, and all these samples were measured within a range of 0.03 to 0.06 micromole per liter. In another area in Greece, Thessalia, where the historical endemic iodine deficiency is more described as very high, there we see averages around 0.02 micromole per liter. A study in Hungary from 1993 showed that nearly 50% of the Hungarian population is supplied by drinking water with less than 10 microgram per liter. So this corresponds to less than 0.08 micromole per liter. So just some other European examples that clearly show that we are under this optimum range for plant nutrition. So if we know that we have a problem or a risk of having too low levels of iodine for proper plant response, we of course started to think as SQM on how to solve this problem. And of course, the first consideration we made was that in order to reach adequate levels of iodine for plant growth, we need to supply it via plant nutrition. Well, SQM discovered that worldwide, the problem of iodine deficiency in irrigation water nutrient solution can be solved by providing iodine together with potassium nitrate, because potassium nitrate is normally used by the farmer in fertigation. SQM investigated and selected a proprietary iodine source which is safe to use and integrated with potassium nitrate in a new product called Ultrasoline. And Ultrasoline K Plus is containing a very specific and controlled amount of iodine, and it's guaranteeing controlled application. In the figure that you see on your upper right side, you do see indeed that in an average use of potassium nitrate, you are able, with applying Ultrasoline K Plus, you are able to reach these iodine levels that are more between adequate levels ranges for plant nutrition. It is easy, adequate, and safe to apply Ultrasaline K+. Indeed, growers can easily apply Ultrasaline K+, just replacing their water-soluble potassium nitrate source that they are used to use. Growers really confirm that for them it's really a no-brainer. They feel safe and they know that just following the programs that they usually use that contain potassium nitrate, where when they just can replace the potassium nitrate by Ultrasaline K+, it's for them really very easy in the management. Potassium nitrate is used in fertigation programs already generally in balanced proportions. So there is already a so-called natural prevention of excess of potassium and nitrogen and even of salinity. And when we, of course, add the iodine in a very specific range to that, then, of course, we will guarantee also to be within safe boundaries with the iodine supply. Just an example from hydroponics. You see on your right side that usually for hydroponics we follow requirements as of the nutrients, the manual nutrient solutions for greenhouse crops. And here we show an example of cucumber in inert substrate. We know quite well what the balance is that we need to provide to the plants. And out of that, we know quite well what the standard potassium nitrate levels are that we need to add in the A and the B tanks. Well, just simply replacing potassium nitrate by Ultrasaline K+ in hydroponics will lead to adequate supply of iodine. Note also that in hydroponics, but also in all other fertigation systems, Ultrasaline K+ has no different effect on the EC and the pH compared to potassium nitrate without iodine. Not only in hydroponic systems, but in all fertigation systems, also for soil-grown crops like drip, sprinkler, or pivot systems, there the same principle is in place to replace the regular potassium nitrate with Ultrasaline K+. I'm just showing here an example of a tomato production in Antalya. You can see in these figures their standard program, which already foresees more than 12 or more than 15 applications of potassium nitrate. A potassium nitrate application to reach 150 to 200 metric tons per hectare, requiring 750 to 800 kilograms of potassium nitrate. Well, replacing this potassium nitrate by Ultrasaline guarantees, on the one hand, that we will supply over many different steps during all growth stages the iodine, and that guarantees that iodine will be available to the plants in all the essential stages where it can be critical. Yeah? And if you look to the overall supply, you see that compared to the control, yes, Ultrasaline applied here is getting you to the range of adequate iodine levels for plants. Summarized, Ultrasaline K+ helps to prevent iodine deficiency in plants. It contains a well-defined amount of iodine. It conveniently and adequately secures iodine supply to fertigated crops. Application with potassium nitrates makes it easy to apply the right dose of iodine. Ultrasaline K+ is produced from potassium, nitrogen, and iodine from our natural resources in Chile. If you want to find out more about the value of our potassium nitrate from SQM, I would like to invite you to our website where you can see more information, amongst other, about, for example, the low carbon footprint that we reach with our methods to produce potassium nitrate. Well, Ultrasaline helps to maximize crop yield and quality and improve resistance to environmental stresses. This, of course, relates to the scientific knowledge that we have been building up over the past years. But of course, it is clear that prior to the launch of Ultrasaline K+, it has been brought into practice, of course, It has been tested in different crops, in different fertigation systems, in different conditions around the globe in more than 100 farm applications. And these farm applications have indeed been demonstrating agronomical value, economical value. And we will now start to give you some insights in some of the results that we have encountered. I would like to give the word back to Katja to talk about some global references.
Thank you, Koen. Well, indeed, you saw the brochure of Ultrasoline K+, and it lists a list of plant metabolism where iodine may be involved in. These are based on the identification of the iodinated proteins, on the unique gene expression of iodine, but also on 100 years worth of publications of iodine and its role and effect on commercial crops. But still, to make sure that the benefits which we predict based on science actually also bring benefit to growers, we did commercial trials with protected crops and in the open field in more than 20 countries worldwide to predict these benefits of iodine as predicted from science for the farmer. For this, we did very simple demonstration trials to make it as easy as possible for the growers to apply the iodine. So we asked growers if they had two different sectors or greenhouses where they could separate the nutrient solutions going to these sectors. In the control sector, the farmer used his normal practice, so we did not ask him to change anything, but the practice did need to contain, of course, potassium nitrate. In the other sector, we asked to keep the planting date, the crop management, the cultivar, and all these aspects the same, but only substitute the potassium nitrate that was used in the control with ultrasoline. So both were applied at the same days, at the same dose, at the same timing. And the only difference between the sectors was the presence of iodine in a micromolar concentration in the nutrient solution. Well, let's give some examples on root growth, for instance. This is an example of lettuce grown in a nutrient film technique, and you see only 13 days after transplant to these production tables, the root growth is much better in the ultrasoline treatment, but also the whole plant weight is higher and the leaves are bigger. So almost 64 grams per plant compared to 24. And what we found in consecutive experiments, we did many in Brazil, that the farmer could harvest these lettuce heads earlier. So he won some days in his production cycle. And if you want to know more about what we are presenting, you're welcome to look on sqmnutrition.com. In the iodine landing page, you will find more details behind these trials. Similar in a trial in Florida, in the USA, in only 6 months of growth of these trees for propagation, and it was orange trees, we found a statistically significant improvement in the root development of these plants. The plant's root system was wider and it contains more lateral roots, so comparable to what we saw in Arabidopsis. Next, the points of root growth, nitrogen conversion, plant growth, and photosynthesis. Of course, these 4 points all have to combine together to result in a high yield of the crop. Well, this picture summarized the 35 best control demonstration trials. So these were trials where we applied ultrasoline right from the start of the crop. We have a well-registered total season yield, and we are sure that the two sectors were comparable. And we used it on a range of different crops, as you see in the pictures. The dark green dots above is the 100% of the yield, which was achieved in the ultrasoline sectors, and the control sectors were consistently behind the ultrasoline sector in all cases. On average, we could attribute 9% of yield lost due to iodine deficiency in the control. This is an example from South Africa, open field tomato in loam soil. And the farmer noticed that the roots of these plants go deeper and are less surface bound. And this corresponded with a modest yield increase of 7.4% by use of ultrasoline. but this translates in 1,600 euros per hectare, which for a crop like this is quite a lot. On the points of stress tolerance, fruit rot, and calcium in fruits and fruit quality, I will start with a slide from a scientific trial in Spain on cherry tomato in sandy soil. Especially in the winter months when the days are short and there is less light going to the crops, we found that in the ultrasoline treatment there were more uniform bunches, the fruits were heavier, larger, but also they had an increased antioxidant concentration in these months. As you see, the carotenoids, vitamins, and polyphenols were consistently higher in the ultrasoline treatments. Well, this indicates that the crop has a better condition despite adverse climatic conditions. And we also measured not only the antioxidants in the fruits, but also the iodine content. And we already knew that from prior experiment, but it was good to see this confirmed in practice, that when you apply ultrasoline, you apply iodine in a micromolar dose, which is sufficient iodine for the crop, but it does not raise the level of iodine in the fruits for consumption. So in both the sector control ultrasoline, in this case, the amount of iodine remained what is normal for tomato fruits, below or just above 2 micrograms of iodine per 100 grams of fresh fruits. We did find a difference in the calcium concentrations in these same fruits. We find consistently, especially during the months of December and January, that the fruits that were treated with ultrasoline had a much higher calcium concentration in the fruits compared to control, which received the normal grade of potassium nitrate without iodine. And again, the rest was exactly the same. So we started to look for calcium concentrations in other demonstration trials, and we found here this trial in Morocco, a raspberry in soil, that with ultrasoline you gained a ton per hectare extra raspberry yields, but also these fruits contained more calcium compared to the control. In Mexico, in blueberry on cocoa peat substrate, we also found an increased calcium concentration in these fruits, but also an increased Brix, so there were more sugars going into these berries. In Morocco, Italian bell pepper, this trial demonstrates why it's important to have your calcium in your fruits. The grower suffered from a problem with blossom end rot in these Italian type peppers. And he noticed that when he applied ultrasoline in one of the greenhouses on his project, he did not encounter this problem in such severe amounts. So in the end, the grower had 10% more yield in the ultrasoline sector, but 14% more yield in the part of the fruits that we could sell for export because of the higher quality of these fruits. Also in the Netherlands, in a greenhouse with tomatoes grown on rock wool with a recirculated drain solution, we found benefits of applying extra iodine in the nutrient solution. This was encountered in 2019 when we had heat waves when temperature outside of the greenhouse reached 40 degrees Celsius. And in 2019, that was seen as exceptional, but maybe this is something we will have to get used to. Anyways, what we saw in these fruits that were collected 6 weeks after the heat wave, so these fruits were in flower when the heat wave struck them, we saw a higher calcium concentration in these fruits, but also they achieved a higher fruit weight compared to the control, which used the normal grade potassium nitrate. And this resulted also in a lower percentage of rot during storage of these fruits of 3 weeks at room temperature. In this same year, 2019 was a good year for us to have trials. We also asked some pepper growers in the Netherlands to try the product. Now, these growers did not have a control greenhouse, so we don't have an exact comparison, but we had feedback from 2 of these growers that actually they had expected their plants to suffer much more during these high temperatures than they actually had. So they asked us if it's possible that iodine can somehow protect the fruit development during heat. Now, we had read in some ancient literature that iodine can be involved in the quality of pollen. In this case, it was apple trees. So we asked ourselves the question, well, does iodine indeed protect the viability of pollen under heat stress? And we asked Professor Gele and Dr. Trin of the University of Ghent in Belgium to do a scientific study to study this phenomenon. What they did is they exposed these little small fruit buds, really flower buds of capsicum pepper, to high temperatures of 35 degrees Celsius for only 24 hours. And then they waited till the flower opened and they analyzed the pollen using a staining method. And with this method, if a pollen stains blue, it's dead. And if it's purple, then it's actually viable. And what they found that if you do not provide extra iodine to these plants, all pollen dies. You have only 3% of these pollen in these flowers which are actually living. Well, if you had fed before, and the treatment started some 6 weeks before we had flowers, if you started before then providing them with iodine, then indeed 25% of the pollen in these flowers still survived. And that would be enough to have a successful pollination. Now, in retrospect, we think that what we hear back from practice from growers, which is mainly about a bigger fruit size, more uniform fruits, a better quality, shine, skin quality of the fruits, well, this may also be related with this property of iodine to improve pollination success. And for instance, this is the case of a muskmelon grown in Thailand in a zero residue crop. And here we had 11% yield increase, and this was solely due to bigger fruits. In Mexico, a trial in raspberry, 1 gram more weight per fruit was found in the raspberries picked from the ultrasoline sector. Now, this may not seem a lot, but if you translate this to the number of fruits which go into a clamshell, and this is an example of the clamshells in the picture on the right, then actually you find you need much less fruits per clamshell, and this translated into a real and measured difference of more than 4,000 more units of sales of these clamshells per hectare. So these are some examples from abroad. And now I give the word back to Koen because he has some experience from Europe to share with you.
Indeed, Katja, a lot of the results that you just demonstrated are really in line with testimonials from growers and trial results that we also see in other places in Europe. Just going to start with the testimonial we got from Poland, where we applied ultrasoline in one greenhouse section, and where we did notice, or where the grower did notice, that in the ultrasaline treatment, the blossom end rot incidences were really nearly absent, while in other sectors of his greenhouse production, blossom end rot was really a big problem in his tomato production that year. This, of course, illustrates also the link with an improved calcium uptake. And although we did not have a complete follow-up of yield and quality parameters, this testimonial in the end struck us. What we did analyze there, though, was the difference in the nutrient solution content as well in the leaf dry matter content in this production facility. And that, of course, clearly illustrates that ultrasaline, yes, is confirmed to supply well the iodine in the nutrient solution, but also in the end, this iodine is taken up by the plant, and it is exactly within the plant where it can fulfill all his roles that are beneficial for the plant growth. In Hungary, we did a more scientifically conducted trial together with our local distributor in his research facility. And there we had, let's say, a rather spectacular results, if you ask me, of 16.8% higher yields. Good to notice that this higher yield, of course, led to a total revenue and economical impact increase. But it's also good to see that especially in the highest quality class, here mentioned in this table as the revenues from class 2, which is the out of class highest quality class. There you see that with a total revenue contribution of 9,000 euro per hectare in the ultrasaline treatment, 4,400 euro came from an increase of fruits in the highest quality class. The increase there was with 49%. Also in pepper in the Ukraine, in open field fertigation in the Kherson area, where we had a plant density of 30,000 plants per hectare and an average consumption of about 500 kilogram potassium nitrate per season. There, the potassium nitrate was applied in the control and the ultrasaline K+ was applied in the ultrasaline treatment, and the results were again higher yield. The relation with the higher yield was mainly found in the fact that we had heavier fruits, about 11% weight increase was noticed. Also nice to see here, if you compare the left side and the right side of the middle graph there, where you see the satellite images on different dates during the season, you tend to see indeed that the vegetation index is better in the ultrasaline treatment, which actually shows that the crop was in overall better condition. In the Netherlands in 2019, and this was the season where Katja already referred to with this heat wave in July 2019 with temperatures above 40 degrees Celsius in many days, which is quite exceptional and hard to manage in the Netherlands. There we did have a partial application with ultrasaline K+, so we only started in the beginning of June. But when we evaluated the results in September, really after this heat wave period, we saw that despite the limited application of ultrasaline, we still had 13% truss weight increase and 12% fruit weight increase. It was also shown that we had improved uniformity and color. So again, indicated that especially also under these stress conditions, iodine has this value. And also related to that, of course, we absolutely wanted to evaluate ultrasaline in Greece and the Mediterranean in general, which also faces challenging conditions related to heat. Here we just had a demonstration of ultrasaline K+, also only applied in the last part of the cultivation, in this case at Hierapitra in Crete. And the farmer, although he mentioned also a yield increase between 6 and 8%, but mainly he testified that it struck him that visually we clearly saw greener, healthier, and more vigorous plants. And especially in periods of high temperatures, he saw less wilting. And that motivated us to have another trial in the same area where we followed up more precisely 2 treatments of each of 5 plants, the control with regular potassium nitrate and ultrasaline. Here again, confirmation of an increase in fruit weight of about 8%. But what was very significant is that in the end, the number of harvested fruits, and now we already evaluated, the trial is still ongoing, 7 different harvest moments, yeah, and we saw that actually the number of fruits per plant was really higher. And when we digged into the details earlier in the season, at 70 days after transplanting, we evaluated actually the number of flowers and the number of fruits. And there we really saw that we had earlier and higher levels of flowers and then of better fruit set, clearly demonstrated here. So We are still continuing in Europe to establish many relations with distributors, agronomists, growers, really to explore value of optimum iodine levels in plants in many fields of interest. I just want to show you a grasp of potential field of interest that we like to further explore or that we are further exploring at the moment. Specifically, conditions creating stresses. Can be cold, can be heat, can be darkness. We have a testimonial, for example, of better flowering after a dark spring in the Netherlands, in pepper. All these kinds of stressful conditions we like to evaluate further, both in high tech as in low tech. And also we like to evaluate if the management response by steering the parameters in the greenhouses really are seen, the responses of the plant are seen quicker, because a well-nutritioned plant, of course, per definition, will react better to management steering by the grower. Other fields of interest are specifically, for example, in crops with attention to root development. Can be the so-called weaker root system crops like strawberry, blueberry, raspberry. We are already also there have indications in strawberry, raspberry, and blueberry that we can reach good results. Also seedlings, propagation, we think that we can really add a good value with iodine. But other crops are not excluded. Iodine is a very beneficial plant nutrient, micronutrient, let's say, in all crops. So we also like to continue to evaluate in fruit trees, oil crops, vegetables like potato, watermelons, onion, etc. So I would really like to invite you to do that together with us. We can come now to a summary and to conclusions. I think we clearly shown in this webinar that science has clearly demonstrated that iodine does indeed take part in plant metabolism and plant growth. And a relation has been also demonstrated with root growth, defense for stress, timely flowering, calcium signaling, fruit production, photosynthesis, sugar production, all for increased biomass. Ultrasaline K Plus offers the most convenient and safe solution to supply iodine to adequate levels for optimum plant growth. The date of this webinar is not by coincidence. It coincides with the launch of the new fertilizer packaging requirements for Europe, yeah, for which means that now from now on, Ultrasaline is ready for commercial launch in Europe, and the packaging of Ultrasaline now will fully comply also to this new requirements of packaging. If you have any commercial or technical requests, please contact us, contact your local representative or your authorized distributor. You can meet us, of course, anytime, but for your information, we will be present in Poland at the Gardeners' Days, Fruit Attraction in Spain, Agrotica in Greece, Indagra, Romania, Agra in Bulgaria, and so on. I would like to finish with the real conclusion. Ultrasaline K Plus is yet another tool developed by SQM to improve yield and quality by optimal nutrition management to increase the revenues of the farmers. Thank you very much for your attention. Now, if anyone would like to add some specific questions to Katja or me, please feel free. We have a couple of minutes left for the webinar environment will close for us. So feel free to ask further questions. Do we have any question here?
Yeah, while we wait for the questions, because I think that we did a good job in answering the questions as they came during the presentation. I do want to comment on the concentration of iodine. There were, I think, two questions on the topic whether or not we are biofortifying fruits. So whether we are increasing the amount of iodine in the fruits enough to solve iodine deficiency in people. And I understand that is confusing because SQM also works on biofortification. So we also find methods to use fertilizers to indeed biofortify, to indeed increase concentration of iodine in fruits, but also in grains, enough to help solve iodine deficiency. What we learned from those experiments is that you need a higher dose of iodine. So you need to provide the plant with a higher dose of iodine than it strictly needs for its own function. So that is as in any nutrient, there is a balance between not getting enough and being deficient, and the getting too much becoming excessive. And for biofortification, you do supply a bit too much iodine for the crop itself, and the result is that the crop moves more iodine into those organs which we eat, because those are organs which for the plant are kind of redundant. It does not need them to grow, it just needs to propagate. If you want to talk to us about biofortification, you're welcome, but it will be another product and another way of supplying the product, because we do not want to provide too much iodine into the human system. So this is why we limit this product, Ultrasol, for use of fertigated crops for crop benefit. And here we apply really the small micromolar dosages of iodine.
I'd like to summarize this clearly. So for human purposes, for fortification of iodine, for human purposes, we leave that to the programs with salt, cattle feed, fortification, and so on. What we focus on is really to optimize the levels in plants, optimize the plant growth to come to better production with higher yield and quality. That must be very clear.
Yes, and in the meantime, I see we have a question of John Constantinidis, and he asks, is there evidence of iodine deficiencies in plants? Meaning, which are the symptoms of iodine deficiency? And that's a very good question, and we get that question a lot. We all know that if you have magnesium deficiency, you have chlorosis, or if you have iron deficiency, your young leaves become yellow. For iodine, we don't have a clear symptom like that. Because as you have seen, iodine plays a role in many different plant physiology systems. So you do not evidently directly see that the plant is iodine deficient. It's just a slowing down and a loss of quality, and less able to steer the crop along with climate, for instance. And also that is comparable to the human system. Often people can be iodine deficient without knowing it. They're just a little bit more tired or have problems concentrating. And this may be related to a slower thyroid function, but it's not something you go to the doctor with. Comparable to what iodine deficiency is in plants. You may not notice, but if you add a little bit more iodine, then the plant will have more energy to boost up its crop production.
This is what is clearly seen in our demonstrations, that you might not notice some deficiency, but if you add some iodine appropriate levels for root uptake, then you see results.
Oh, I see a question that there was some confusion about the time of the webinar. Maybe that has to do with global time changes. You can see in the sticky poster that after this webinar, you can get on the email a link, and you will be able to replay the full session. So you can watch us again if you would like to, and possibly you can also share this link with other people. But I'm not sure about that, how that works with the registration. But at least all people that registered will be able to replay the session. Also, if you want to learn more, you can connect with the SQM Nutrition website. We have an iodine landing page. It's above in the menu, and there you'll find more information about iodine, but also the trial which we've done with. And also, if you click on the files section, you see there are a number of brochures that have been shared with you, so you can go those.
So I would like to invite you for further questions. Please do send us an email directly or to our general email address that is mentioned in this presentation, or contact your local representative who will pass on these questions to us, and we will be happy to come back to you.
I think we are perfectly in time, Koen. We did not expect it. We were expecting to be way over. We did a very good job.
I did expect this. But maybe because in this webinar environment will at a certain moment in time close down. This is what this technology does. results. So before that happens, I would really, of course, like to thank everybody for your attention.
And I see that our email addresses are in the chat box, so please note them down. And if you have more questions, you can send it. I see one more last question of Sufyan about the index of salinity, potassium nitrate compared to ultrasoline. And that's also a very good question. Thanks for bringing that up, because actually there is no difference between potassium nitrate with or without iodine, because we add only 0.1% of iodine to the potassium nitrate. So the rest of the product is identical. So we do not expect any changes in EC or pH other than you would normally have when you mix potassium nitrate in your nutrient solution. And also the foliar question that comes in, well, we still have time to answer that. The product was developed, used in fertigation. This is what iodine for, and this is what we defined our concentration in potassium nitrate for. Foliar application, we have investigated, as I said, we work also on the biofortification. So we investigated the uptake of iodine in plants, and we know that plants can take up iodine from a foliar spray solution. But what we still need to find out is actually how much iodine you need to apply to get a benefit for the crop. So thank you very much for your attention, and I think we will close it now because the hour is up. And
we will come back, and for sure also on the foliar questions we have some comments to align with you. We will do that as soon as we have the chance to talk to you.